Go ahead and start.
Okay, welcome to today's sustainability seminar. My name is Nancy Holm. I'm Assistant Director here at the Illinois Sustainable Technology Center, and I'm the seminar organizer along with Beth Mashesky, who's sitting up here manning the computer. Our next sustainability seminar will be at noon on Thursday, November 8th. We will have a presentation on the microbe water subsurface porous media interactions given by Revere Singh from the University of Illinois here in Urbana-Champaign. So please plan to join us for that talk on November 8th. Today, before we begin the seminar, I'd like to ask everyone here to please silence your cell phones if you have not done so already. And then also, we're going to hold all the questions for the speaker till the end, and then I'll come around with the microphone so we can record those because we are recording the seminar today. And for those of you viewing online, you may type your questions in at any time, and we'll answer those at the end also. So today I'm very pleased to welcome as our speaker Vishal Verma. Dr. Verma is an assistant professor in the Department of Civil and Environmental Engineering here at the U of I. His current research focuses on assessing the health impacts of ambient air pollutants. Dr. Verma earned his BE in Environmental Engineering from Delhi College of Engineering, his Master's of Technology in Environmental Science and Engineering from the Indian Institute of Technology in Bombay, and his PhD in Environmental Engineering from the University of Southern California. Dr. Verma has earned numerous awards and recognitions for his work, including the Gold Medal for Outstanding Research Performance in the Indian Institute of Technology in Bombay, the Verdi School of Engineering Fellowship Award, and the Outstanding Researcher Award from the University of Southern California. He's an active member of the American Association of Aerosol Research and is chair of its health-related aerosol working group. He's also a member of the International Society of Exposure Sciences and the American Geophysical Union. In addition, he's a reviewer on numerous journals related to environmental science, air pollution, and health. So please join me in welcoming Dr. Verma as our speaker today.
Thank you, Nancy, for such a nice introduction and inviting me here to present my work. So I'm assuming that people here are at least concerned about the air pollution, but just to express my feeling, about this problem is this is the picture I took in 2016 when I was in here last time. You can barely see India Gate, and the same year I read this news on CNN that New Delhi was the most polluted city on earth that year. So the topic of my presentation is oxidative properties of ambient particulate matter, and in this work I'm going to present last 11 years of my research in which I tried to assess the relative contribution of various aerosol or the particulate matter components in the oxidative properties. So I will start with a brief background in which I will basically talk about the major routes of urban aerosol formation and the oxidative potential measurement, like the definition of it, various mechanisms to explain that process, and also how do we measure it. Then I will talk about the various sampling campaigns I have been involved in, for example, in the Western United States while I was doing my PhD work, and then Southeast United States while I was doing my postdoctoral work, and finally the Midwest US where I'm currently working. So in all of these sampling campaigns, the major focus was to identify the specific components of the particulate matter and their emission sources in the ROS or the oxidative potential of the particles. For example, in the Western United States, we tried to find out the bulk fraction of the particulate matter which were associated with the oxidative properties. In the Southeast US, while I was doing my postdoctoral work, we tried to further narrow down the specific components which were related with the oxidative potential, and in the current work which we are doing at Midwest US, we are trying to develop new methods and newer probes which actually combine the various actions of different components into a single measurement. So starting with the major routes of urban aerosol formation, The exhaust of the vehicles driving on our freeway and street consists of both gaseous and particulate components. In the gaseous components, we have these gases like sulfur dioxide, carbon monoxide, nitrous oxide. And in the particulate component, we have the solid particles like elemental carbon and metals. Some of the precursors of the particulate matter, for example, these organic and inorganic substances are also emitted in the gaseous forms because they are very hot at the exit tailpipe of these vehicles. But once these cool down, due to atmospheric dilution, and if the conditions are favorable, they can actually form tiny particles called the nucleation mode particles. So after cooling down, they can form this nucleation mode particle, or if a big droplet has already been formed, then this nucleation mode particles can just condense onto it to form even a bigger droplet. Or if there is a surface available in the atmosphere, then these nucleation mode particles can just condense onto it to form this externally mixed aerosol. Or the other route could be that these nucleation mode particles can just link with the solid components like elemental carbon and metal to form these fractal-like structures, which is commonly known as soot. So what happens during the aging process, particularly during the afternoon period or during long-range transport, that these volatile particles can again evaporate due to atmospheric dilution or higher temperature, and then can mix with this originally emitted gases and vapors. And once everything is in the gaseous form, various photochemical oxidants, for example, ozone and hydroxide radicals present in the ambient atmosphere, can attack on them, and in presence of sunlight, it can oxidize them. We know that the products of this oxidation reaction generally have larger molecular weight because of the addition of the molecules, so they generally have lower vapor pressure, and they can again come back to this particulate form by condensation, or by condensing onto the solid particles to again form this externally mixed aerosol. Thus, based on the formation mechanism, the sources of the ambient particulate matter in an urban atmosphere can be classified into two sources: primary sources, which include these vehicular emissions or industrial emissions or biomass burning, and the secondary sources, which include these various photochemical oxidants and the reactions. So why do we worry about this ambient particulate matter? Various epidemiological and toxicological studies have shown the strong links between the concentration of this ambient particulate matter and health effects. Perhaps the first study which established these links very concretely is known as the Harvard Six Cities study. The study has been cited approximately 10,000 times as of now. What the authors showed very elegantly in that study is that the concentration of these fine particles, ambient PM 2.5, in 6 American cities was perfectly correlated. I mean, it's almost unbelievable with the adjusted mortality rate ratio in that city. I stress the word adjusted here because the rate ratio shown on the y-axis was actually adjusted for various risk factors like smoking or other lifestyle factors. So the rate ratio shown here is truly the mortality attributed to the ambient PM 2.5 or the fine particulate matter. I mean, after that, there have been many studies, in fact, the follow-up study, which actually reassessed the data developed by this study just to confirm this relationship. And all they found, I mean, there are at least 10 papers just to on the reaffirmation of the data developed generated by this study, which all actually showed the same results. So the links between the ambient particulate matter and health effects are well established. But what are the mechanisms? Like what is the mechanism by which these particles actually cause these health effects is still a matter of debate. A relatively new hypothesis which has emerged in recent years is that when these ambient particles are inhaled in the human body, they deposit at various places depending on their sizes. For example, in the upper respiratory system, we have this bigger particle deposited. The smaller particle goes to the deeper region, and the smallest particle, called the ultrafine particle, goes to the deepest region of the lungs, known as the alveolar region. In fact, the deposition of these particles in various parts of the respiratory system has been well studied, and the exact fraction of this different size particle in a specific region of the lungs can be accurately quantified by various models available. So the hypothesis linking these ambient particulate matters and health effects suggests that wherever these particles are deposited in the respiratory region, they interact with the various antioxidant and the lung and the cellular structure of those of that lung, and in that interaction, an extremely reactive group of species known as the reactive oxygen species is generated. Once these reactive oxygen species or ROS are generated, they basically initiate a series of chain reaction, and it basically, I mean, they are so reactive that they start actually oxidizing anything coming to their pathways, for example, DNA, proteins, or lipids, which ultimately lead to the cell death and eventually translated into various diseases such as cardiovascular or the neurological disorders. So the question arises, is ROS generation a foreign phenomenon to the body, or is it completely natural? And to get to that point, we will have to understand the basic mechanism of the aerobic respiration, as it turned out that this is the most basic process of energy production in all living beings, which is inherently associated with the ROS generation. So in its simplest form, the equation of the aerobic respiration can be written in the form that glucose is oxidized by oxygen, and in the process carbon dioxide and water is generated, and energy in the form of ATP molecules are released. But the actual process is much more complicated than this. I'm not going to go into the details of it, but just briefly describing here that glucose first undergoes glycolysis to form pyruvate, which is then degraded to form acetyl coenzyme A and carbon dioxide. This acetyl coenzyme A then enter into the Krebs cycle, where which happens in the mitochondria. And in this Krebs cycle, the two major products of this reaction is the cellular reductants known as NADH and FADH2. So the main purpose of this reductant is to donate electrons, which are then transported through this complicated electron transport chain, at each step of which a fixed number of ATP molecules are released. The most important point to be noted here is that the last molecule which accepts this electron is oxygen, which in presence of hydrogen ions, protons, it actually forms this water molecule. So this is the normal or the basic process of the aerobic respiration. But as we know that nothing in nature is absolutely perfect, so is true with this process. What happens that in 0.1% of the cases, these electrons are preternaturally accepted by oxygen, and in the absence of proton ions, hydrogen ions, it directly accepts this electron and forms the superoxide radical. So this is the first step of the ROS production in the living beings. Once the superoxide radical is generated, it initiates a series of other chain reactions by which it starts accepting other electrons to form first hydrogen peroxide and then hydroxide radical. So it's basically a whole cascade of the ROS formation in the cells, by which it starts forming more and more reactive oxygen species. So if ROS generation is a natural phenomenon to the body, then the question arises, then there must be a cellular defense mechanism to protect itself from ROS. Otherwise, the cell basically will just be died up by the natural production of the ROS. And there actually is. Every cell has a pool of antioxidant. It can be of different types, but they're all known by their capability to donate electrons. So whenever an ROS or a free radical is generated in the body, which is characterized by this free electron, unpaired electron, the antioxidant donate their electrons to free radical and expands it and stops it to oxidize other components. So what is the problem? We have the ROS and we have the antioxidant. In fact, there is no problem in a healthy individual where the concentration of the antioxidant is perfectly balanced by the concentration of the reactive oxygen species. The problem arises when, due to some reason, the concentration of the reactive oxygen species exceeds way above the capacity of the antioxidants to neutralize them, or due to other reason that the concentration of the antioxidants depletes to such a minimum level that even the normal ROS generation cannot be supported. So both of these processes both of these conditions are known as the oxidative stress, because the cell undergoes the stress created by too much oxidant, which cannot be neutralized by this pool of antioxidants. Okay, so what is the role of our ambient particulate matter in creating this imbalance of the reactive oxygen species and antioxidants? Let's look at the formation of the superoxide radical one more time. That oxygen, as we learned, that oxygen by accepting electron form the superoxide radical. Generally, this electron is donated by a coenzyme, for example, NADH or FADH2. But this electron is not directly accepted by oxygen, but rather it is first accepted by a compound, let's just call it R, which that gets reduced in the process, and then it donates this electron to oxygen and gets back to the original form. Thus, in this process, this compound behave in a cyclic manner, which keeps on taking electron from coenzyme and keep transferring to oxygen and forming the superoxide radical. Such compounds are known as the redox cycling agent, and this is exactly the role played by this ambient particulate matter. That we know that there are certain components in the particulate matter, and that is our research question. What are those components which are known to behave in this redox cycling manner? I should mention that there is another process of ROS generation in the living beings, and this process is more complicated. We know that certain biological cells, for example, these macrophages, when they come into the contact of this ambient particulate matter, they identify them as foreign substances, just like an allergy, and then when in an attempt to engulf or oxidize them, it's a biological process that they generate this huge concentration of the reactive oxygen species. And this process is more complicated in a way that it basically involves so many biological processes like cellular signaling or other processes. But certain studies which have tried to find the link between this biological ROS generation and this chemical ROS generation have found this link that the redox cycling properties of the particles actually are linked with this biological ROS generation as well. Okay, so researchers have developed various methods to assess this ROS generation capability of this ambient particulate matter, and broadly, they can be classified into two categories: cell-free method and cell-based method. In the cell-free method, what we do is that we try to simulate the conditions of the cellular environment in a chemical system, basically taking the similar pool of antioxidant present in the cells and then incubate it with the particulate matter. And then what we do here is either we measure the rate of consumption of these different antioxidants, because higher is the generation of the ROS, you see that the more will be the consumption of this antioxidant, or directly measure the rate of formation of the ROS. So various methods available under this category is diethyl assay, ascorbic acid, which is the vitamin C present in our cells, and glutathione assay, which is another major group of antioxidant, and this respiratory tract lining fluid, which is basically a mixture of various 4 antioxidants present in our lung lining fluid. In the cell-based method, we use specific cells, for example, macrophages or epithelial cell lining, and then expose these cells to these particles by first extracting these particles in a suitable medium and then putting these cells into that medium. And then we measure the actual damage done to these cells as an indicator of the ROS generation capability of this particular matter. So we use macrophage ROS generation assays as well as the epithelial cells lining assays in this category. So with this introduction, I would like to introduce the overall research questions we actually investigated in my research group, which is focused on these 3 questions. Is mass of the particulate matter, which is currently used regulatory parameter to control this aerosol pollution, also a comprehensive measure of its toxicity? If yes, then we don't have to be worried about. We can still keep using this particulate mass as the regulatory parameter, right? But if not, then we have to ask the second question: that are there specific components in the particulate matter or their mixtures which are more efficient in generating the ROS or causing this oxidative stress than others? And if so, then the third question arises: from where they are coming? What are their emission sources? Could it be nuclear sources? Could it be biomass burning? You know, in Southern California last year, and there was so much biomass burning. So what is the most important source which is actually generating these components in the particulate matter? Why do we need this? Because that's how we can actually better control them rather than just focusing on the other sources. So to accomplish this objective, we collect particles from different emission sources. For example, we collect the particles directly from the exhaust tailpipe of the vehicles, which are known as the primary particles in a dynamometer facilities. These primary particles, we saw in one of the previous slides that these oxidize in the atmosphere and then form secondary particles, which we also collect and then see what is their potential to generate this ROS. And finally, we collect particles from biomass burning. And from all these particles, what we do here is we measure the specific chemical composition of these particles, for example, organic carbon, elemental carbon, metals, inorganic ions, and also measure the capability of these particles to generate reactive oxygen species. And then, by statistical techniques, we try to find out the linkages in the ROS generation capability of these particles and the various chemical components. That's the first step of our approach. That we try to first find out by statistical analysis what are the components which could be associated with the ROS generation. The second part of our approach, which I call the mechanistic analysis, we try to break apart these particles into various subcomponents like elemental carbon, inorganic ions, organic and metals, and then we try to see what fraction of these particles is most associated with the ROS generation. So by removal of these different components, how much is the effect on the remaining ROS activity of the particles? As in a way, we try to apportion this ROS generation capability of the particles to various subcomponents like these inorganic ions or elemental carbon. So we use, during my PhD and postdoctoral work, I use basically these two different probes to measure this ROS generation capability of the particles, which include both acellular method, meaning the chemical method, and the cellular method. In the acellular method, the name of the probe which I use is known as the DTT or the diethyltritol probe. What we do here is we take up this compound DTT, which can be which can be classified as a generic reductant present in our cells, representative of this aryl group of antioxidant present in our cells. And we incubate this compound with the aerosol particle. And what happens that certain aerosol components we know that actually can oxidize this compound DTT while releasing these electrons. So, and these electrons are then transported by this aerosol component to oxygen, which then form the superoxide radical. So you can see that in this method, it basically simulates the same process as occurring in the cellular mitochondria, which is like by the formation of the superoxide radical from the NADPH. So what we do here is we measure the rate of consumption of DTT and we assume it to be proportional to the rate of formation of the superoxide radical. So that assay is commonly known as the DTT assay. In the macrophage ROS assay, we use this rat alveolar macrophage cells, and in those cells we inject a dye called DCFH. And then we expose these cells to the aerosol components like, where these particles and then extract them in a medium and then put these cells into that medium. And then these particles generate reactive oxygen species in these cells, and these reactive oxygen species once generated in the cells, so this dye is inside the cell. This ROS can actually oxidize this dye DCFH to DCH. Now the difference between DCFH and DCH is that DCH is fluorescent. So once the DCH is generated in the cell, it actually fluoresces the whole cell, and by counting the fluorescence intensity of the cell, the whole plate method, we can actually see what is the rate of formation of the reactive oxygen species by this particular method. So we have applied these two methods of measuring the ROS generation on the samples collected from different emission scenarios, different emission settings, and compared the response. So the first comparison I'm showing here is for the vehicular emission versus biomass burning. So in summer of 2007, the year I came into the United States, there was huge wildfires in Southern California, and immediately I grabbed that opportunity. Basically, the smoke you could see all over in the Los Angeles basin, and we collected these particles, these biomass burning dominated particles from the ambient atmosphere. And a few days later, when this wildfire was subsided, we got these particles dominated mostly by the Wickler emissions. So basically, I applied these two methods of measuring the ROS generation into these particles, and what we found that the DTT activity, meaning the acellular method of measuring the ROS generation, showed a consistently higher trend for the biomass bonding particles than the Wickler emission. So you can see that the DTT activity was consistently higher for the biomass bonding than the Wickler emission. But when I measured the ROS generation using the cellular method, using the macrophage ROS generation assay, we found there is no consistent trend. Sometimes it is higher for the biomass burning aerosol, while sometimes it is higher for the Wickler emissions aerosol. Okay, so now I switch here to another comparison. In summer of 2008, I collected particles in two different time periods of the day, morning period versus afternoon period. In the morning period, particularly in the Los Angeles basin, you can clearly see a signal from the Wickler emissions, which is much higher in the Wickler in the morning period, and that these primary particles which are emitted from this Wickler emission, by the afternoon they get aged in the atmosphere and then they form the secondary particles. There are many studies which have previously shown that the various chemical signatures, that these are the secondary particles and these are primary particles. So what we did is that we compared this the response of this acellular method of ROS generation using the DTT method, and what we found, you can see here that the secondary particles showed a much higher response in the DTT assay. So the DTT activity is is consistently higher for the secondary particles than the primary particles. But again, comparing the response of the cellular method of ROS generation, you can see that sometimes it is higher for the primary particles, but again, sometimes it is higher for the secondary particles. So basically, there is no trend in the cellular method of ROS generation, but for the chemical method, is much more reproducible. You can see that there is a trend, right? So why is it behaving like this? So that led to a certain hypothesis that that there is something inside the particle which could be associated with the emission sources, but not necessarily, which is probably driving the response of this different ROS generation mechanism than just the emission sources. And with that hypothesis, we measure what is there inside the particle, the various chemical components, right? So we measure these different chemical components in the particle, and we try to basically do a very simple statistical analysis, simple regression, and what we found is that the concentration of the water-soluble organic compound, which is known as the water-soluble organic carbon, was highly correlated with the response of the acellular method of ROS generation with the DTT, which is the DTT assay. But the cellular method of ROS generation, which is the macrophage ROS, can be explained in terms of the concentration of few transition metals. You can see the high correlation between R square reaching up to 0.9 by these different metals. So basically the cellular ROS is probably more driven by these different metals rather than the organic compounds. So based on this, this simple statistical analysis, we have some idea about the different components in the ambient particulate matter, that is the water-soluble organic compounds and metals, which are probably driving the response of this different ROS generation mechanism. But as you all know, that there are limitations associated with that statistical analysis, right? I mean, irrespective of how good or bad correlation could be, it does not necessarily prove the causation, right? So to further confirm and quantify the effect of these different components, we tried some mechanistic analysis in which we remove these different components and see the impact on the residual ROS activity. So in this slide, I'm going to explain our different mechanistic techniques which we use to basically remove these different components and see the effect on the ROS activity. So the first technique, which is used to remove the organic compounds, basically we use a thermodenuder. This thermodenuder basically consists of two sections, intersection and the absorber section. So in the heater section, these organic components are volatilized, and once in the vapor form, we have the secondary section, which is the absorber section. So these volatile components are absorbed into this section. So what we have coming out from the thermodenuder is non-volatile particles, which is free from these volatile components or the organic compounds. So it's basically the particulate matter minus the organic compounds. The second technique for removing the transition metals, we use this diesel exhaust particle and we pass them through a Keyless column, which contains this immunodiacetyl ligands. And these ligands binds efficiently with the transition metals. So we pass the aerosol straight to this Keyless column, and what we have coming out from this column is a PM extract which is free from all the transition metals. So now we have this, PM component which is free from another component, right? Kind of. So we applied this. We measure the ROS generation activity of these segregated PM components, and here I'm showing you is basically that the effectiveness of our mechanistic technique, which is used to remove the organic compounds, the thermodenuser. And you can see that this technique is really effective in removing the organic compounds. As you can see that more and more we heat the aerosol, the higher and higher removal is for the organic compound, right? And interestingly, similar is the effect we see on the DTT activity, which is the acellular method of ROS generation, which is removed approximately by the same amount as the organic compound, right? At 50 degrees Celsius, removed about 40%, and then at 200 degrees Celsius, about 70%, right? So based on these results, we have some confirmation that organic compounds are probably playing a major role in driving the acellular method of ROS generation, which is measured by this DTT assay. Okay, now for the second mechanistic technique, which is the chelation technique for removing the metals. So you can see that this technique is also very effective in removing various metals. For example, the most metals of concern like manganese, iron, they are removed to an extent of about 80 to 100% by this chelation technique. And interestingly, again we see a similar impact on the cellular ROS activity measured by this macrophage ROS assay, which is removed approximately by the same amount, around 80%, by chelating the PM samples. So again, based on this technique, based on these results, you can say that these transition metals are probably the major driving factor in the cellular method of ROS generation. Okay, so that was the work presented during my PhD work, research. The research always generates more questions than you work on, So it's basically, three major questions arise from this work. The first question could be, if ROS activity or the oxidative potential of the matter of the particulate matter is an important property of the particulate matter, then why don't we start analyzing it on a regular basis like the other components like particulate mass, or the other components like organic carbon or elemental carbon. The problem here is, there are two major concerns, time and labor. Just to give you one example, while I was doing my PhD work, we used to send these samples to UCLA for analyzing this ROS activity by the DTT method, and you basically send 5 samples. It would take basically a month, to get this results back. And when I visited that laboratory, I realized that it basically requires 2 people to analyze 1 sample, and it basically takes like 3-4 hours to get the data for, using the DTT method. So it's really time and labor intensive approach. So the second question could be, okay, we find out that water soluble organic compounds and metals are the major species which are associated with the ROS generation. But unlike metals, water soluble organic carbon is not a single species. It's basically a collection of many organic compounds which are grouped under this water soluble organic carbon, right? So there could be millions of compounds present in this water-soluble organic carbon. So the natural question arises, can we further narrow down this specific component in the water-soluble organic carbon which are really associated with the ROS generation rather than this associating with the bulk fraction, right? And if we can identify these, then the third question is, can we really identify the emission sources of this ROS active components? These questions look different, but you can see that they're linked to each other. For example, to find out the specific component, to find out the emission sources, first we need to find out the specific components, right? Then only we can find out from where they're coming. And to basically apply any technique to, to do this source apportionment using the emission for the emission sources, we have to have generated a large data set, right? And that there's a constraint associated with the generation of the large data set is the time and labor, right? if the one sample requires 4 or 5 hours to analyze one sample, how are you gonna generate this huge data set? So with these questions in mind, I moved to Georgia Tech in 2011, and the first thing we discussed with my postdoctoral advisor is that can we actually automate this whole process of measuring the ROS activity in a simplistic manner? And we came up with this instrument, which we published in 2013, the paper on, that basically we just developed this automated instrument. It consists of 2 syringe pumps, the big and the small syringe pumps, which can be controlled by this, easily through a computer. And the purpose of this pump is basically they take various, reagents from various reservoirs and do the reaction in this reaction vial. And then after reaction, basically they take out different liquids and send it the product for the to the spectrophotometer for the absorbance or fluorescence measurement for the ROS emission. So what we have to do here is just collect the particles, extract them in a medium, and then put them into the sample queue, which consists of either 7 or 14 samples, and then just forget. I mean, just put the samples in the overnight, and then by the morning they will all be analyzed. A similar system we developed here at Illinois also, which is in my lab, which is operating, and I mean we have made actually many modifications to the system now. In fact, last year, based on this automated system, I mean this system can actually analyze the ROS activity of the sample which has already been extracted. So you have to even collect the samples using a filter and then bring it to the lab and then extract it, right, and then put it in the automated system. In this new instrument which we developed last year, we coupled it with a real-time sampling device to basically generate the real-time data on the ROS activity of the particles directly collected from the ambient atmosphere. So basically, this system is coupled with this online mist chamber. It directly collects this PM 2.5 from the ambient atmosphere, and then these particles are collected into this mist chamber into a PM suspension, which is then fed directly to this automated system to provide this online measurement of the ROS activity. So we tested the system for like 50 days in last summer. And based on the data, we were able to produce this first ever recorded of the diurnal profile of the DTP activity of the particles at an ambient site, at an urban site. We are currently investigating. I mean, I'm not going into the details of it, but we found out that this tri-model pattern is actually associated with the different emission sources at the site currently. Anyway, so having developed this system and knowing that we can now analyze the ROS activity of the ambient particles on a much larger sample size than which is previously impossible, we launched our intensive sampling campaign in Southeast US. Basically, what we did here is we installed our high volume sampler at various sites. For example, Jefferson Street site was the representative of the urban Atlanta region. And then Georgia Tech site, so we put our sampler on the rooftop of our building, and then one sampler on the right besides a major freeway, which is known as the roadside site, I-75-85 in Atlanta, and one site in the rural area, which is about 70 kilometers west of Atlanta. So on all of these sites, we collected this PM 2.5 Leiden high volume filter, and on a fraction of that filter, we measured the concentration of elemental and organic carbon. The remaining filter we punched in the smaller, we punched out smaller cellular punches and then extracted them in water to obtain this water-soluble extract. On this water-soluble extract, we measured the concentration of matters and water-soluble organic carbon, ground carbon, and the ROS activity. The remaining extract was passed through a C18 column, a solid phase extraction column. The beauty of the C18 column is that it actually separates this PM components into hydrophobic and hydrophilic fraction. I will again come to this hydrophobic philic fraction because we found out later that turned out to be the most important aspect of our study. So in this hydrophilic fraction, we measure the concentration of matters and ROS activity, and by difference of the hydrophilic fraction and the total fraction, we assess the ROS activity associated with the hydrophobic fraction. Okay, so the C18 column. So a conventional C18 column looks like something like this. So this is the column and this is the PM extract. So when we pass our PM extract to this C18 column, the hydrophobic stuff, which is brown in color. So the hydrophilic stuff actually, is directly passes through the column, which is a colorless liquid. But the hydrophobic stuff, which is brown in color, it is written on the column. So various researchers have tried to find out the chemical composition of this hydrophilic and hydrophobic stuff, and what they found, this hydrophilic stuff is mostly consists of aliphatic or the long chain, hydrocarbons, and this hydrophobic stuff is basically associated with this large molecular weight aromatic compounds, which are also known as hulies, because they are very similar in nature in the chemical composition as the humic substances present in natural water and soil. So humic-like substances or hulies. That's the name abbreviation given to these compounds. One good thing about this column is that matters also directly pass through this column and are collected in this hydrophilic stuff. Thus, this technique of separating the hydrophobic and hydrophilic stuff can be used to segregate the contribution of the organic compounds and matters in the ROS activity. And we know that these in our previous investigation, we found that these are the two major groups of species which were associated with the ROS activity. Okay, so the first problem was solved of the time and labor, right? We can analyze the samples on a we can analyze many samples for the ROS activity. Now you remember the second question was narrowing down the specific components of the organic aerosol which are associated with the ROS generation, right? To answer that question, we use this recently developed this instrument by Aerodyne company, especially $700,000 instrument. This is known as the high resolution aerosol mass spectrophotometer. So we installed that sampler at this spectrophotometer at many sites. The beauty of this instrument is that it actually can segregate this organic aerosol data into various sub-fractions. For example, hydrocarbon-like organic aerosol, which is the primary organic aerosol, which is directly emitted from the vehicular emissions, and isoprene-derived organic aerosol. If you go to southeast, there are many pine trees there. It's all covered with the forest, right? So those pine trees release this volatile organic compound isoprene, which is oxidized in the atmosphere to form this isoprene-derived secondary organic aerosol. Biomass burning organic aerosol, then oxidized organic aerosol, which is a generic secondary organic aerosol, and then organic aerosol from cooking activities. So we have this chemically segregated organic aerosol data, and we have this ROS activity measured on our high volume filters. So the first thing we did is we tried a simple linear regression between this different chemically segregated organic aerosol data and the ROS activity, and the components which were significantly associated, meaning high R value, then entered into this multiple linear regression model where we tried to predict the ROS activity of the particulate matter in terms of the concentration of this different organic aerosol, plus residual, which the residual could be anything like the metals or which were not included in the model. So the linear regression model relating the ROS activity and the chemical component takes this generalized form, right? Now the important thing in this equation is that if the unit of ROS is in ROS generation, which could be nanomole or something, per meter cube of air, and the unit of this organic aerosol is in microgram per meter cube of air, then the unit of this coefficient has to be in terms of the ROS generation per microgram of organic aerosol, right? And in that unit, it basically represents a mass normalized, which could also be known as the intrinsic property, right? Intrinsic property meaning it doesn't depend upon the substance, right? So it's basically it becomes intrinsic ROS activity of that organic aerosol, and this is what I show here. So by that model, we estimated this different intrinsic ROS activity of the various organic aerosol, and interestingly, you can see, which is also somewhat surprising, that biomass burning organic aerosol has the highest intrinsic ROS activity, which is almost 3 times the that of the diesel exhaust particles. So we think that diesel exhaust particles are the major culprit, but we found out that the intrinsic activity of the biomass burning organic aerosol was 3 times higher than these diesel exhaust particles. Cooking organic aerosol, right? Cooking organic aerosol also has substantial activity, right? But we were not very sure of this estimate because you see the large error bar. And then oxidized organic aerosol also has some significant activity which is similar to the digital exhaust particle or the ambient particles. And then isoprene derived organic aerosol, which is the biogenic aerosol, from the pine trees, doesn't seem to have very high intrinsic activity. Okay, so this was the intrinsic activity, right, which is the mass normalized activity of the organic aerosol. Now, if I multiply this intrinsic ROS activity with the actual mass concentration of this different organic aerosol, what I will obtain is the total ROS activity of the organic aerosol, right? And this is what I show here. So the sum of the different mass is basically the total ROS activity from the organic aerosol at that particular site, right? So now you remember that we also measured the ROS activity on the hydrophobic and hydrophilic fraction separated by the C18 column. So what I did here is we compared the measured ROS activity on the hydrophobic fraction of the organic of the ambient aerosol with the modeled ROS activity of the organic aerosol, and what we found is a nice comparison between the two at various sites. I mean, there is sometimes off, but it's mostly comparable. What does that mean? So it's a little complicated concept, but I would try to explain it in a very simplistic manner. So in an ambient particle, we have two major groups of ROS active species: organic compounds and metals, right? So what we did in this analysis is we segregated the PM components into hydrophobic and hydrophilic components. And we know that in the hydrophobic, from the chemical composition data, we know that in the hydrophobic fraction, we have mostly aromatic organic compounds, right? And in the hydrophilic fraction, we have aliphatic organic compounds and metals, right? So what we see in this comparison is basically that the ROS activity, the model ROS activity, is almost equal to the measured ROS activity on the hydrophobic fraction. So what does that mean is, whatever ROS activity is coming from the organic aerosol is basically coming only from the hydrophobic substances or the aromatic substances, and the aliphatic organic compounds do not contribute anything to the ROS activity. I don't mean to say that the hydrophilic stuff doesn't contribute, because hydrophilic contains metals also, right, which might be contributing. But because this total ROS activity is equal from the organic aerosol is equal to the aromatic ROS activity, basically that means that only aromatic compounds are contributing, right? And you know that this hydrophobic substance is also known as ULIS. So we basically narrowed down a specific group of the organic aerosol known as ULIS, which is driving the ROS emission mechanism in the organic aerosol. And if you remember, that was our second question, right? Now coming to the third question, the emission sources of this different organic aerosol, of the organic or the inorganic components contributing to the ROS activity. We basically used advanced receptor modeling technique known as positive metric factorization. I'm not going to go into the details of it, but we basically applied that model into a large data set of ROS activity and the chemical composition data. And basically we developed this time series of the different emission sources in the ROS activity. And you can see that almost all of the sources, emission sources, contributed to the ROS activity of the particles. But there is strong seasonality, right? In the summer, we have most of the ROS activity coming from the secondary sources, that is secondary sulfate and secondary water soluble organic carbon, contributing to the ROS activity. But in fall, this is a mixed contribution, but it's still, from all the sources, but still the highest contribution comes from the secondary sources. And interestingly, in the winter, all of the ROS activity is dominated by the biomass burning sources, so which is contributing to about 50%, right? So the liquor sources also contribute significantly to the ROS activity, but its contribution, you can see, doesn't change a lot during different seasons. It remains around between 10 to 20% in different seasons. And finally, the dust also contribute to the ROS activity, but very minimally, around less than 10% generally. So that was all, my PhD and postdoctoral work. So now in the next couple of slides, I'm going to show you what we are doing currently here at Illinois. So the questions we are exploring currently here is, so we started with the slide that can we actually segregate the contribution of these different components. But now we are thinking, can we really segregate this contribution of this organic and the metals in the ROS activity? Why I'm asking this question is, which none of the researchers actually previously thought about, what if there are interaction between these components, which could be both synergistic and antagonistic interaction, right? And if so, they could be actually altering the ROS activity in a very complicated manner so that we can not really segregate the contribution. And the third question is, if there are interaction between the organic compounds and metal, do we have the appropriate probes or the methods to capture this interaction? For example, the DTT assay is one of the most commonly used or the most traditional probe available in our field to capture this ROS activity. But does this probe also capture the interaction among the PM components? We don't know. So just to explain, this last point again, in a more clear manner. So you know that we already discussed about that oxygen. What is the ROS cascade of the in the cell, right? That oxygen by accepting an electron from the superoxide radical. So this superoxide radical by accepting one more electron can form hydrogen peroxide, and this hydrogen peroxide, in presence of metallic ion, for example iron, can form this hydroxide radical, and you know that this reaction is known as the Fenton reaction. So this is the ROS cascade. Now this is the second step of the ROS cascade. The question is in the most traditional probe, the DTT probe, do we capture this second step of the ROS cascade? And the answer is no, right? Because what we measure as I explained earlier, what we measure in the DTT assay, we basically measure the rate of consumption of DTT, which we assume to be proportional to the formation of the superoxide radical. But what this superoxide radical is doing in the downstream reaction, we don't know, right? If we just measure the rate of formation of the superoxide radical, we won't know that what is the rate of formation of the hydroxide radical depending on the presence of this metallic ion, right? So this is the basic limitation of the traditional ROS activity probe, the DTT activity probe, that we were not capturing this whole cascade of the ROS emission. And this limitation could be the reason that why you remember that in the that we found out that the only organic compounds are more contributing to the DTT activity and not the metallic ions, because the metallic ions are involved in the second step. They are not covered into the first step, right, measured by the DTT assay. So to capture this second step, we developed this new probe last year. What we did here in the DTT assay, we actually added another compound, DPT, disodium terephthalate, and this compound actually reacts with hydroxyl radical and becomes fluorescent. So by measuring the fluorescence intensity at 425 nanometer wavelength, so it can be excited at 310 nanometer excitation wavelength, we can measure the rate of formation of this hydroxyl radical. So now the newer probe we have, we basically measure both the DTT consumption activity as well as the rate of formation. So which is the rate of consumption of DTT is proportional to the rate of formation of the superoxide radical. And then we directly measure the rate of formation of the hydroxide radical. So the first step is probably more dominated by the organic compounds. We know that like certain organic compounds like quinones or PHs actually drive this first step. And the second step is driven more by the metallic ions. So by capturing both first and the second step, we are basically incorporating the individual contribution of these different components as well as the interaction between them. But do we really capture that? And here are the results. So you can see that in what we did here is basically we made a mixture of iron and phenanthroquinone, and we measured both the DTT consumption activity as well as the rate of formation of the hydroxyl radical in the DTT assay. And you can see that the effect is mostly additive in the DTT consumption, meaning that the quinone are actually able to, they are contributing somewhat to the rate of formation of the superoxide radical for the DTT consumption, and similarly the iron. So the effect is only additive. But look at the results for the rate of formation of the hydroxyl radical. The mixture always has a higher activity than the sum of two, right? So basically indicating the synergistic effect. Why do we have the synergistic effect here? So like quinone are actually, go back to this one here, quinones are driving this first step very efficiently, right? Generating the superoxide radical. When the iron presents, this iron actually can take the superoxide radical or the hydrogen peroxide and then form the hydroxide radical. the mixture of this quinone and iron actually has a much higher response than the sum of two. So basically what we are trying to say here is that by measuring the second endpoint, we are able to include the contribution of the individual contribution as well as the synergistic interaction between these different components in the ROS activity. We even extended this interaction experiment to the real ambient organic aerosol collected directly from the real PM samples, which is known as the Hugh-List, with different methods, and you can see that DDT assay has some interaction. I mean, this is the interaction factor. So if it is 0, that means there is no interaction, but if it is higher or lower, then there are synergistic and antagonistic interaction. So there are some interaction factor in the DDT assay also, but in the hydroxyl radical generation in the DDT assay, these interaction factors are much more pronounced, right? Sometimes it is very synergistic, sometimes very antagonistic. So, okay, so the newer probe is actually capable of measuring of this individual as well as the interaction between these different components. But what is the biological relevance of it? Do these reactions happen in the biological system also? So for assessing that, we are collaborating with another faculty at Illinois, Michael Peva, and what we did here is basically we basically are trying to assess the toxicity of these particles using these Chinese HAMS low-risk cells. So, we basically take this particulate matter fraction from the filter and then extract them in a suitable medium and then expose these Chinese HAMS low-risk cells. And the endpoint we measure is a long, complicated procedure, but the endpoint here is the LC50, which is the lethal concentration of the mixture or the sample that kills 50% of the cell. So remember that lower is the LC50, more toxic is the sample, right? So we compared the response of this different traditional and the newer probe with the LC50, and you can see that the traditional probe is related with the is correlated with the LC50, right? R is like around 0.58. But look at the R value for the newer probe. It jumped from 0.58 to 0.82. So basically, what does it mean? That the newer probe is capable to incorporate the individual and this interaction among the PM components in the ROS activity measurement is more biologically relevant than the traditional probe, right? Okay, so with these expertise and the newer, facilities and the capabilities of measuring the ROS activities for the ambient particles, we have launched our intensive sampling campaign in the Midwest US. So what we did here is basically we have launched we have set up our samplers at various sites in the Midwest US. For example, we have 3 samplers in the Champaign. One is on the in front of the Beckman Institute. Another one is just right across the building at Illinois State Water Survey, and the other one is actually on the Bondville site, which is classified as a river site. And then 3 samplers in surrounding cities, one in Chicago, Indianapolis, and St. Louis. And what we are doing here, we are basically collecting weekly filters from all these sites. And the plan here is that to continue the sampling for a year and measure the ROS activity of these particles by various assays which are available till date. So we are not limiting only to the DDT assay, but some other assays have been developed recently by other researchers at other universities. For example, lung lining fluid assay, where we measure basically the rate of consumption of the ascorbic acid or the glutathione consumption or the actual ROS generation, and also the macrophage ROS assay, and also measure the cellular toxicity using the live cells. For example, we can measure the alteration in the gene expression and also the reduction in the metabolic activity, so that we can answer this question if ROS activity could be a proxy for the cellular toxicity of the aerosol. And also we are measuring the chemical composition of this different ambient aerosol, meaning that elemental carbon, organic carbon, water soluble organic carbon, brown carbon, basically to find out what are the chemical components which could be associated with the cellular toxicity. And then by comparison of this chemical component and toxicity and ROS activity and toxicity, we can see which among these probes is actually best able to represent the contribution from these different components in the PM to basically represent the cellular toxicity. So with that, I would like to acknowledge the various funding agencies which have funded this work, US EPA and Health Effects Institute, and the wonderful group of students I have at Illinois, Jinnai, Joseph, Varan, Shishang, and Varanya. Thank you.
Thank you very much for your talk today. Very interesting. Do we have some questions here in our audience? None so far. Beth, do we have some online questions? Okay.
Based on my understanding, like a DDT kind of the test is the more sensitive than the cellular kind of the test.
So it is more sensitive?
Yeah, more sensitive or the more the matchable to the like.
I mean, you cannot say it's more sensitive. It's basically it means that it is driven more by specific components than the other probe, which is probably driven by other type of components. So, I mean, we cannot say one probe is more sensitive than the other. It's just a different set of components probably playing a major role in that probe than in the other probe. I think that's what we can say. I mean, same is true for the other chemical probe also. I mean, I didn't go into the details of it, but ascorbic acid, people what they found that it is driven by one component in the particulate matter, which is copper, and then similarly the glutathione consumption. So that is the caveat about these different probes, that they are probably sensitive to a specific set of the components. And I think what we need here is a probe which actually combines the individual as well as the synergistic or antagonistic interaction among the PM components to basically represent the overall ROS activity of the aerosol.
So, and the second on the minor question is, can you specify like aliphatic compound?
Basically,
like aliphatic compound.
So,
what's the water soluble aliphatic?
So it's basically like the simple linear carbon, which is not the ring form structure. Those are the simple aliphatic species. Yeah, I mean the aromatic compounds which are ring formed species, they are written on the C18 column, so they are the aromatic species. So.
I'm just curious that is kind of something more aliphatic compound.
They're what? Sorry. Yeah, that's very true.
Good talk. So, question is for the organic aerosol. The source will be,
Sorry.
You have a lot of types of the organic compound in the organic aerosol. Also, source will be different. For example, in California, but you most focused on urban. But California also have a large agricultures. As they know, at times they're using a lot of fumigant, a lot of chemicals. They can cause the ozone. I think the based California EPA, they mentioned as like a agriculture contribute like 45% ozone generation. So that's why I concern. But in the Illinois, we don't use any fumigant. So that's why the organic, organic aerosol, the components will be different from California. Illinois, also Arizona. You mentioned in the the George.
That's very true, yeah.
Should be organic components should be different.
Right.
So that's my question is the cost of the ROS, should be different.
Right. Yeah, definitely. In fact, I mean, your question is very relevant because we found that in the Southeast US, the biomass burning was the major contributor to the ROS activity, but probably not true for the California. Maybe, I mean, there are wildfires in California as well, but maybe not true for Illinois because here you have an entirely different chemical composition of the organic aerosol. I won't say entirely different, but it could be somewhat similar. But yeah, it could be different. I agree with that. I mean, in fact, I remember that the clear pattern of the primary versus secondary particles we see in the Los Angeles, we didn't see that in Georgia. I mean, there is no primary or secondary signal. It's basically a very mixed signal because it is. From the results, we see that it's mostly the secondary organic aerosols which are driving the ROS emission. The secondary organic aerosol, they might have a background impact. Like background source. For example, they are coming from the long range transport, so which doesn't have a clear, morning or afternoon signal. So I mean, you're right that it could be, and that's why we need to conduct these studies in different parts to see really what is the driving factor in this region. I mean, for example, in India or China, it could be very different organic aerosol. So yes.
So in earlier part of your study, you showed that water-soluble organic compounds and metals, those were the key components that were giving you those RO effect. But in the end, you showed that there's no contribution of WSOC, water-soluble organic compound. It's mainly metals and aromatic compounds. Is that right?
Yeah, but the aromatic compounds are part of the water-soluble organic compounds. That's the thing. So that's a little confusing because you see that the PM method which we pass, how do we measure this aromatic compound? We pass this water-soluble PM extract to the sputum column, and the fraction which was written on the column was known as the Hewlett, which was mostly abundant in the aromatic fraction, aromatic compounds. So I mean that is hydrophobic fraction, but it's not water insoluble. It's relatively, so in the PM extract there is a range of the hydrophobicity. It's not that when we say hydrophobic compound, that doesn't mean the water insoluble. It's a whole range of the hydrophobicity. So it is hydrophobic, but it is water soluble. So when given the chance between to partition between the sputum column or remain in the hydrophilic fraction, it will prefer to remain on the column. But it is basically an aromatic substance and it is water soluble. So the fact here is that the water solubility is a complicated issue, right? I mean it could be very less, but the amount of water we use to exchange these particles, and it's basically very similar to what the amount of the lung lining fluid which is present in our cells, right? It's actually much larger than the concentration of these particles. So with that amount of liquid available, even the slightest soluble component could become soluble, into the water or the suitable medium. So
did you also look at the effect of rain, like what happens to the air quality after that? Because I assume that after rain, most of that water soluble organic compounds or those kind of components will be washed and made.
So after rain, yeah, that is true, but we didn't see the effect of it. I mean, the rain has a sweeping effect. Basically it moves. I mean, it has a larger impact on the coarse particles, the big particles, but it has some effect of the smaller particles also. I mean, these organic acids are mostly in the smaller range, generally. These coarse particles, the big particles between 2.5 micron and 10 micron, they are mostly metallic particles. So like, or the inorganic substances. By the very, mechanism of the formation, these organic particles are generally concentrated in the smaller sizes. So rain definitely will have some impact, but I don't think it will remove these particles completely. So.
Do you have any questions here? Okay, with that, we'll end today's seminar. We thank you again
Thank you so much.
for your presentation.